and in basically conjunction with the American Society of Human Genetics conference, gnomAD version 4.0 was released. And so as we thought at the SPC that all of you, or almost all of you, are heavily using this treasure trove of data, we thought what we should have as a plenary, the lady of gnomAD herself. So please welcome Caitlin Samosha.
Good afternoon, Berlin. I'm incredibly excited to be here at ESHG to discuss our most recent release of the genome aggregation database, gnomAD 4. We had a 4.1 release very recently that we strongly suggest you use. So as a conflict of interest, I do receive some funding from Microsoft for unrelated rare disease diagnostic work. I will not be the first to say this, and I will not be the last to say this, but to properly interpret the variation that we see in any individual's genome truly requires studying genomes of hundreds to hundreds of thousands of people. It takes understanding the variation that we see in the general population. And I would argue that we need large and diverse reference populations to do that most properly. And there are many, many reasons that these can be helpful. Two that we'll focus on here are, of course, improving variant classification and interpretation. In particular, when it comes to rare disease diagnostics, being able to rule out variants as too common is a really critical aspect of having these reference populations. And we know that databases like gnomAD have contributed to diagnoses around the world. The second is studying the patterns of variation within the populations. Where do variants occur? Where do variants not occur? Understanding that allows us to improve our understanding of mutational intolerance, and it allows us to understand how the forces of selection have shaped our genome over the eons. Of course, there are many other important applications here, such as informing drug discovery, or if you have phenotypic information, tying changes in the genetic information to changes in phenotype. But these are the two we'll highlight more today. So this is an excellent time to be a human genomicist, and that's because there are increasingly large collections of sequencing data available for reference populations online. I usually show this lot with many more data sets on it from a while ago, but I've had to restrict it to those data sets that have over 100,000 sequenced individuals, and this is a mix of genomes and exomes. But you can see we're up to almost 1 million exomes or genomes being publicly available. So we have the TopMed data set, our prior release of gnomAD, the genome aggregation database, V2 had about 141,000. Of course, the All of Us study from the US is still actively sequencing. The UK Biobank we'll hear about quite a lot at this conference, I'm sure, but they're a major resource. And our recent release of gnomAD version 4.1 had a little over 800,000. I should also point out that Regeneron Genetics Center had a 1 million exome data set, which is about 822,000 unrelated individuals that you can also browse online. I want to take a moment here to draw your eye to the colors on the screen here. This is identifying individuals by inferred genetic ancestry group. So we are really trying to cluster individuals based on genetic similarity, and we use provided metadata labels to give these high-level data names here, excuse me. You'll see there's quite a lot of this lightest blue bar on this plot, which indicates individuals of broadly defined European genetic ancestry. And they are massively overrepresented in all of the data sets on this slide. This is not globally representative at all. Now, some of this comes from the fact that the largest data sets here, gnomAD v4 and the Regeneron Genetics Center 1 million exome data set, are using UK Biobank, which was collected in the United Kingdom. However, I also want to point out that there are groups that are underrepresented here. So Middle Eastern genetic ancestry, for example, is in gnomAD v4. There's only a couple of pixels on the slide right now, so we understand that that's a data set that's underrepresented here. There are other populations that are completely missing. For example, populations from Oceania currently aren't listed on this plot at all. So this is to say that these resources are powerful and important, and they can still be helpful, But your mileage may vary if you're studying, for example, a patient from a population that is not well represented in these data sets. And just to say this isn't about identity, this is a genetic similarity clustering metric. So focusing in on the genome aggregation database, or gnomAD version 4, this is about 731,000 exomes and 76,000 genomes that were all natively aligned to GR-CH38. We got there. They were also jointly processed together, a really extensive quality control, and we take that quite seriously because we do know that this database feeds into diagnostic pipelines around the world, and we do our best to put out the best data set possible. Of course, this doesn't happen from a single individual lab or even a single center. This is from 100 studies from around the world with 308 data contributors. And almost all of the samples in here are coming from case control studies of complex adult onset diseases and from biobanks. So that means that this is not a perfectly healthy set of individuals because no one is perfectly healthy. This is supposed to look like the reference population. And so especially as we see more and more biobanks coming online, if someone is healthy enough to make it into a biobank, you will see their variation within these data sets. We do try to remove related individuals, however, so you know that there's no over-representation of variants based on that. In terms of our sex breakdown, it's about 50-50 XXY individuals, and the median age is in the 50s, 54, 55. So we do want to emphasize that as this data set has grown about 5 to 6 times from our most recent releases, which is on here, we are seeing fold changes of 2 to 10x for different genetic ancestry groups. And within this most recent release, we have 3 times as many individuals of inferred non-European genetic ancestry. So that works out to be about 170,000 of them, which is larger than the entire V2 release that many of the tools and metrics we made were based on in general. So while we understand that this is still not great representation in terms of the global diversity we see in the world, this is still a massive increase in the number of individuals that are in one of these publicly available data sets. So since this is primarily exomes at this point, of course, the bread and butter data that we have included are single nucleotide variants and indels. However, at the same time that we released the single nucleotide variants and indels, for version 4, we also released copy number variants, so deletions and duplications. We, of course, have genomes, which you can get SNVs and indels from as well, but for them, you can get a lot of other types of variation. So we released structural variants at the same time. Deletions and duplications are included in that, but also more complex structural variations such as insertions and inversions. We have called short tandem repeats on a subset of our genomes, and they're available online for about 60 disease-associated loci. And we do have mitochondrial variant calls from the genomes as well. So there's quite a lot of data within this database across a number of different variation types. So what are we seeing? We have over 900 million SNVs and indels, which works out to about 2 SNVs every 3 base pairs. This is about 10 million synonymous variants, 16 million missense variants, and 2.5 million predicted loss of function variants in this data set. I'll try to refer to them as PL throughout the rest of this talk. We are starting to approach saturation, but mostly for highly methylated CpGs that lead to a synonymous variant. So methylated C right next to a G is quite easily deaminated. It's one of the most mutable contexts in the genome. And we can see 99.8% of all possible synonymous CpG substitutions when it's a highly methylated site. We do see slightly lower numbers for missense and for loss of function. And I don't want to oversell this, though. This is the most mutable context. We are nowhere near saturation for the other contexts within the genome. But we are approaching it for this particular one. We did, again, concurrently release copy number variants and structural variants. The data set includes roughly 67,000 rare copy number variants from the exomes. This is a site frequency of less than 1%, which is done for quality control purposes, which works out to about 31,000 deletions and 36,000 duplications, about 1 per person. This is work from Jack Fu. And so that means you have about 1 gene disrupted per person from these rare exonic CNVs. We also have about 1.2 million structural variants from the genome samples themselves. So about half had not been previously reported in any of the other contemporary studies. This works out to about 12,000 per person. and they result in a loss of function in roughly 188 genes per genome, though of course the genes that are impacted by these are those that are more tolerant of being lost. So that's depicted over on the right of the slide, and I should say that we have a talk on this on Sunday at 10:30 from Zhufeng Zhao, who's leading this structural variation work, and she's going to dig into more of the non-coding aspects of this as well, because that's one of the exciting things about having genomes within the data set. That's just a little bit of intro to what we have. How can we actually leverage some of these large population data sets? Again, I'm using gnomAD as an example, but all of us in UK Biobank and other examples will have the same properties here. So the first, of course, is frequency filtering. This is one of the most important use cases. It is particularly important in diagnostics. You have a patient, you have a variant that you think is a good candidate, and you want to know if it's been seen in other people in the general population, specifically if it's been seen too commonly. Now, there's a lot of work out there about where exactly you should draw that allele frequency threshold. However, I think many people would agree that variants with an allele frequency greater than 0.1% can often be removed, except in certain cases. So in the gnomAD v2 data set, where we had roughly 140,000 individuals, we had about 500,000 variants with an allele frequency greater than this 0.1% cutoff. So that's 500,000 variants around the exome, actually, since this is mostly an exome data set, that you can immediately remove. They're being colored here by which genetic ancestry group that variant was found in above that allele frequency threshold. And you can see there's contributions from all of the different groups. But as we move to v4, the proportion of rare variants from European samples hasn't increased at all. In fact, let's do that again. You can see that even though we've massively increased the European samples, this has stayed the same. So in fact, the number of rare variants per sample, actually more common variants per sample, is lowest for the European genetic ancestry group. And some of this is we're starting to hit numbers within the European genetic ancestry group where we have a pretty good sense in the coding region of what is a common variant and what can be therefore removed from these diagnostic pipelines. But there's much more to learn about some of the other populations around the world. So this increased diversity allows us to rule out variants that are too common because they're too common in one genetic ancestry group. So if we take, for example, a variant that is considered rare in the European population, I have given you another arbitrary allele frequency threshold of 0.01%, so rare in the European genetic ancestry group. The question is whether it's also globally rare, or whether it's not so rare in some other non-European genetic ancestry group, and therefore can be potentially ruled out. And I should say, variant classification is far more complicated than this. Variant rarity is one aspect, but it's not everything. So these variant of unknown significance and likely benign are dramatic oversimplifications here. So we wanted to look into that comparing v2 to v4, and what I'm showing you here are missense and loss of function variants that are rare in the European genetic ancestry group, so again, that allele frequency cutoff, but they are more common in a non-European genetic ancestry group, so they're being colored here, again, by the group that they're more common in. So this, again, in the gnomAD v2 data set, we had a little under 500,000 variants that could be ruled out due to this. But as we jump to v4, we see that there's over 200,000 more variants that can be ruled out because of the increased ancestral representation in this data set. So again, sample size is important, and I like data. More is better, more is more, but we do know that it's not just a sample size only, it's also who's being included within the data sets. So with that, we'll move a little bit into variant curation, which is really going to trip on my favorite science mantra, which is that the more interesting something looks, the more likely it is to be false. So you should really take that to heart in your life. It's something I think about all the time. But this holds for when you're looking at these population data sets. If something looks like it shouldn't be in there, maybe it's not. So to walk through this, we first wanted to focus on ClinVar variants that were present in v2 and v4. And this is work being spearheaded by Sanna Gudmundsson. It's unpublished coming out of Anna O'Donnell-Lurie's lab, but we're hoping this manuscript will be up soon with some follow-up by Sarah Stenton. So in gnomAD v2, we saw about 56% of all ClinVar variants were in that v2 data set. Mostly that was coming from benign and likely benign variants, and it was a little more rare for the pathogenic. So only 16% of pathogenic, likely pathogenic ClinVar variants were in the v2 data set. But as we move to V4, we see this jump. In fact, the number of likely pathogenic ClinVar variants in gnomAD V4 is 30%, so this has doubled. And in total, you see it's about 73% across the board. However, when we dig into this a little bit more, we do see that most of the pathogenic variants have lower allele frequencies in V4 compared to V2 when they were both present, so that's at least nice to see. We got a little lucky or unlucky, however you consider it, in our first wave. But we really wanted to see what was going on here. So Sana was looking into the known inheritance patterns for these variants in ClinVar, and we see an average about 7.2 pathogenic alleles per person, but almost all of them are associated with autosomal recessive inheritance, which is what we would expect. So on the plot on the left, she's comparing the inheritance of ClinVar variants, likely pathogenic in ClinVar compared to gnomAD. And in gnomAD, you can see an expansion of this yellow bar for autosomal recessive, and really a shrinking of those associated with X-linked or autosomal dominant inheritance. But they're not completely absent. So as a follow-up to some of this, we're trying to understand where these are coming from, how are they getting into our data sets, and we are seeing, for example, pathogenic variants in dominant genes associated with severe pediatric conditions. So for example, KMT2D associated with Kabuki syndrome has 13 variants in gnomAD V4 with 41 alleles, which you would potentially not expect to see in our data set. When we dig into this a little bit, almost all of them are coming from biobanks, such as the UK Biobank. Shouldn't necessarily be a surprise that it's the major contributor since it is a big chunk of the data set of gnomAD V4. But just to emphasize here, if someone is healthy enough to get into one of these cohorts, they're ending up in our data set. But there's also this additional factor of incomplete penetrance or maybe some subthreshold effects, and there's a session on that on Tuesday morning if you'd like to know more. Sana then did a deeper dive into 77 haploinsufficient genes associated with severe conditions, and specifically 734 PL02.2 variants within these genes, to understand whether we should be seeing them within our data set or not. So she applied a loss-of-function curation framework that was published on an AJHG last year that evaluates whether a PL02 variant has been rescued by a secondary sequence property, such as a multinucleotide variant, whether it has an uncertain biological relevance, for example, falling into the last exon or falling into an exon that's lowly expressed. And it also evaluates potential technical artifacts, strand bias, homopolymer runs, things that would make you, where it's not really a real variant. So of these 734 loss-of-function variants in haploinsufficient genes, 70% of them have evidence that they are not actually resulting in a true loss-of-function variant. So again, you're seeing it in the data set, but it might not be having the effect that you would initially predict just based on, for example, a VEP annotation. So this is, on the left, what's depicted as some of the reasons that those variants did not seem to be, that evidence that might not be a true loss of function. Quite a lot of last exon or low PEX, that's that expression score contributing, but you do see frame restoring indels contribute to a number of them, and of course, there's a combination of explanations that contribute to a lot. So the 70% that have evidence of not resulting in a true loss of function here for this set, by contrast, when you compare it to heterozygous PL02 variants in 22 autosomal recessive genes, you see only 27% of those have some kind of evidence of not being truly loss of function, or 29% from a set of homozygous loss of function variants found in the gnomAD V2 data set. So that manual curation I just mentioned was done in gnomAD V2. Again, the homozygous loss of function variants or the heterozygous loss of function variants in recessive genes, we have about a little under a third have some evidence of not being truly loss of function. But as soon as you shift to haploinsufficient or more dominant genes, it's a very different story. So we should say we do have this curation information up on the browser. So when we've done it, we're trying to share with the community to let you know that we're not so sure that this is a true loss of function variant. And all of these are in silico approaches. Obviously, multiplex assays of variant effect might be able to sort of nail this down with even more certainty in the future. Finally, of course, we can use the patterns within this data set to understand selective constraint. And really what we're looking for here is to identify genomic regions that are intolerant of mutational changes. There are two primary approaches here. So the first is looking at a shift in the site frequency spectrum from common down to rare. And the second, which is what we've been focused on within the gnomAD consortium, is looking for a depletion of variation within the human population. So you might expect to see a number of variants there, but you're really depleted. So this is a signal that we've been looking for. It does, of course, require that you have some sense of what you're expecting to see. So we have a mutational model that works fairly well here. So when we compare the expected to observed number of rare synonymous mutations per gene, we see a pretty tight correlation. That's an R of 0.983. And each of these points are a gene. So for the vast majority of genes, synonymous is matching. As you move to missense and loss of function or PL02 variants, we start to see a little bit of a drop off that line, specifically for PL02. So missense here is actually also pretty tightly correlated. But this depletion of PL02, where essentially every gene has fewer loss of function variants than we'd anticipate, we believe to be the action of selection in our genome. Now, we've come up with multiple measures of loss of function and tolerance. The slightly older one is PLI, which is a dichotomous metric. It's supposed to be sort of a yes-no. However, more recently, we've used LUF, or the loss of function observed over expected upper bound fraction, which is a continuous metric, and it is supposed to be pronounced as the egg in French, but my French is awful. So one of our chairs can kindly correct me here. And just to highlight here, this is a statistical bound on the observed over expected, just because we know that gnomAD is a sample of the general population. So one of the issues with these approaches is that PL02 variants are rare. Not only are they rare allele frequency-wise, there aren't that many opportunities to create them in a gene. So that we know that when we applied these approaches to prior data sets, there were a number of genes that we were underpowered to detect constraint in. I'm happy to say that in gnomAD v4, we are powered to detect constraint against PL02 variants in 93% of the genes in the human genome. This is compared to V2, which was about 72%, or XAC, which was under 50%. So as our sample size grows, we're doing even better at being able to measure this intolerance. In terms of comparing constraint scores, we just ran the exact same pipelines on V2 and on V4. We see very similar performance when it comes to the LUF rank. So if you're looking at using LUF, we often decile it. We see that the most constrained LUF deciles are enriched for haploinsufficient genes, while olfactory receptors are in the least constrained LUF deciles. Autosomal recessive variant genes get a spread across the board. I should say that if you'd like to know more about how this was applied, the comparisons between V2 or V4, we have a poster on this led by Kristen Lurikia. So on Sunday, please go talk to her about how this has been applied to this new data set and its performance. We also have other constraint metrics. I should say I'm a little biased. I really like the gene level ones. I've been in exomes for a while, but there's other things within our genome to look at. So we do release non-coding constraint scores. These are Z-scores from a NUCI, genomic non-coding constraint of haploinsufficient variation. That's depicted both on the region view and on the variant page. We have regional missense constraint up, so this is looking for pockets within genes that are particularly intolerant of missense variation when you might not see it across the gene. I forgot to label it, but that's BAP1. It's on V2 for now, but obviously we're going to be updating that. And we are working with Nicole Lake's team at Yale to bring in mitochondrial constraint metrics. So we won't only have this for observed over expected for mitochondrial genes overall. Nicole has some really fabulous regional missense constraint for the mitochondrial genes, which we'll be displaying. But what's coming in the future? I've mentioned a few features. We know that there are more features that were in V2 that need to be updated, such as variant co-occurrence, so some statistical phasing work, and our regional missense constraint metric, but we're always focused on bringing more samples. So I mentioned earlier that we understand that gnomAD could do a better job of representation, so our V5 data set is going to be focused on including the over 400,000 diverse genomes coming from the All of Us next release. And Angie has a poster on the All of Us data set and all of the fabulous stuff they have in it, which I believe is on Monday. So All of Us itself, their release, I believe, is going to come out in the next year, and then we will quickly incorporate it. So we've already started conversations. But looking more to the future, we'd like to create a federated gnomAD. So the idea here is that every center or country will process their data according to similar quality control that we have in-house. They can stand up separate browsers, but we'd want aggregate allele frequencies and quality control information shared with the primary gnomAD for ease of community use and improved constraint metrics. And we've already started some of these conversations with countries around the world and with EGA. So this is a little bit of a call to action. If you have one of these data sets and would like to be part of the federated gnomAD, we are looking for partners right now. And while I have your attention, we're running a user feedback survey. We care a lot about how the data set's used, and we want to make sure that this is the most useful for the public. So if you have feedback, you can go to broad.io/2024_survey. And with that, I'd like to wrap up by saying that gnomAD version 4.1 has over 800,000 exomes and genomes. Obviously, allele frequency filtering is a critical use case, but you should be aware of things in the data set that look like they shouldn't be there. And finally, measures of selective constraint. We've re-released them in version 4, and we have more coming in this space, so keep an eye out for it. Of course, this happens with a large team, so I'm showing the core gnomAD team there, some of the main data contributors, but if you'd like to know more, you can go to our About page. Thank you.
Question for Caitlin. Yes.
Hi, thank you very much for all the data in gnomAD, first of all. So one of the things that we frequently want to do is, as you say, you have inferred ancestry for all these data, but sometimes you want to be a bit more specific and have like a closer vector space, well, the vector space of all these variants and be able to match our samples much closer to some of the ancestries. Is there any plan to have that more fine-grained representation of ancestry?
So we do release our PCA loadings, actually our LD matrices. We have been considering a way to try to match into gnomAD based on samples' genetic ancestry. That's something that's under development, so I don't want to give you too much promise here because we're still trying to make sure it works well before we release it to the general public.
Great.
Yep.
Please.
Thank you so much. gnomAD is obviously an amazingly important resource, and it's great to see this progress. But my question is also around ancestry. So there is underrepresentation. I think it's actually much worse than those images that we're all showing because we have one group for African ancestry. That's actually not showing how big the genomic diversity in that group is relative to everybody else. My question is, are there better ways of grouping people into ancestry groups that would actually improve diagnostics in everybody, because some African groups have a lot of genomic diversity, variants that seem rare in other groups that we could filter out, but these people are either not assigned to African ancestry or are merged in a gigantic group of other African groups. So could you improve ancestry assignment?
So we're currently running, we did a local ancestry inference pipeline on the admixed American genomes that's currently being run on the African ancestry genomes as well. I believe that should be out hopefully later this year or next year. So you're right, we know that there's some improved representation that can happen there. We're also trying to work with African continental groups to be able to bring in more individuals of African ancestry from Africa, because almost all of the African ancestry individuals present in our database or in all of these databases are coming from individuals from the US and the UK, so there's biases in what variation from Africa was sampled.
Yes, thanks very much. Great talk. I'm here on your left. and above all, great job. Karl Revolta from University of Basel, Switzerland. I am a medical geneticist, and I use gnomAD a lot to exclude occurrences of homozygous variants in genes, disease genes that have yet to be discovered. However, there are some, as you were saying before, some patients in version 4. And it's a bit of a bummer for us because we cannot know whether we should expect these people to be there or not to be there. My question is, in case you have knowledge of individuals being patient, are you planning on flagging these genotypes, specifically homozygous genotypes?
So we try to do the loss of function curation just to tell you whether something looks real to help out with that information. For phenotype, frequently that's not available to us and or we're not allowed to release that information. So we understand that that's a frustration. A major benefit is that UK Biobank and all of us allow you to go check their own data sets for some of those phenotypes. So again, we should be depleted for severe pediatric conditions, but incomplete penetrance is a thing, and we are seeing some of these individuals that look relatively healthy in some of these biobanks without the associated phenotypes. So it's a little bit, it's challenging on both sides. We have removed any cohort that was recruited specifically for a severe pediatric phenotype, such as autism. But otherwise, that's probably something we'll be unable to do, I'm sorry.
So for the sake of time, we'll take one question, and I please ask Stylianos and Jean-Louis to come after the session to Caitlin. So one last question.
Thank you very much for your very important work, and I have a very short question.
Yes.
In version 2, you had a variant co-occurrence or phasing tool that you don't have in version 4. I think it's not there. So
We're working
will
on
this
it.
come, or are you planning to not?
We're working on it. We're trying to get, this is a heavy lift to get all this data set out, and it was critical for us to get allele frequencies to the population before we had all of our tools back in place. We're interested in that, and we've already started to bring in some of the copy number variant and structural variant information as well. I can tell you, for example, that copy number deletions on one side with a loss of function variant on the other side don't occur that often. So you're still not seeing a kind of double knockout from CNV's loss of function, SNVs, which we were able to show SNV in our prior work. But we understand that that's a resource that people are interested in having.
Thanks.
Yep.
Thank you, Caitlin.